So again, now this is the rationale for BAB study. This is just the definitions. Bioavailability, as we know, bioavailability is the rate and extent to which the API or the active moiety that we call active pharmaceutical ingredient is absorbed from a pharmaceutical dosage form and becomes available in the systemic circulation. That is what is bioavailability when we mean. And bioequivalence is two pharmaceuticals are called bioequivalent if they are pharmaceutically equivalent or they are pharmaceutical alternative and their bioavailabilities. So how do we measure bioavailability? In terms of rate, which is Cmax and Tmax, and its extent of absorption, which is area under the curve. After administration of the same molar dose under the same conditions are similar to such a degree that their effects can be expected to be essentially the same. So that is important that when you are doing bioequivalence study, so dose has to be similar. Like you can't compare a 100 milligram to a 150 milligram or a 200 milligram. So whenever you are doing a study, so doses, etc., have to be similar. I mean, we'll come to that later, that how you have to standardize your study, what are the factors that you have to take care while designing your protocol, designing your study, so that you achieve the bioequivalence.
If we want to go for a bioequivalence study of a dosage form like 10 mg, say for 10 mg, yes. If there is no formulation in RLD available for 10 mg, then can we go for 2 mg 2 tablets?
Yes, in many cases people do that, but in that case, it will not be a ANDA. I mean, if you are even if it is a new dosage form also, then also regulations changes slightly. I mean, for 10 milligram, if your dosage is 10 milligram and there is not a reference product available, reference product is of 20 milligram. So yes, you can design your study by giving 2 tablets of your 10 milligram this thing. For lower, I think for lower strengths it is not a problem. But suppose your product is say 20 milligram, and there is no reference of 20, reference is 10. In that case, when you are going in for higher dosage, so you might have to convince the regulatory about safety aspects also, because you are going in for higher strength. Yeah. So those questions are there, which you have to take care. If you are going in for a dose, you can design the study by, I mean, giving 2 dosage or like that. But what is the rationale why you are going in for a higher strength, that all has to be there as part of your dossier.